Welcome to our session on identifying and solving common pitfalls in flow cytometry data analysis. This is intended to be a very interactive session, so if people can come forward, we've got a small group in a very large room. If we can move forward or consolidate to some tables, I think it'll be a little bit more fun.
And maybe try to have at least one table buddy. We got some overachievers down front.
And this is also the point of the session where I would go over the housekeeping notes, but as per my discretion, I don't think any of these apply. You are welcome to take pictures if you're so inspired. Don't think we have any confidential information. So you're welcome to do that. And we don't want you to hold questions to the end. This is intended to be a conversation and discussion starter. All right, as people are rolling in, I just want to get a sense for our audience. A lot of familiar faces here, but who regularly does flow cytometry data analysis? Who does data analysis but of a different flavor? Oh, okay. And who deals with people who are doing their own analysis? All right. How many of you guys are core managers versus technicians? And then technicians. All right, so we have a very diverse group, actually. The goal of today's session is to diagnose flow data analysis when it has gone wrong. And we would love to get your feedback on your thoughts on that, as well as we'll showcase some examples of when data has gone wrong that we can all diagnose together. I'm Alice Wiedeman. I'm from Benaroya Research Institute in Seattle, Washington. There, I manage the human immunophenotyping core. I've been working with CyTOF in high-parameter flow cytometry for about a decade and have had a lot of experience with people struggling with data analysis in that field. I'm joined today by our other speakers, Caroline Rowe and Dr. Celine Lages. You can see their pictures up there. Mix and match. Caroline and both are highly experienced in flow cytometry analysis and facility operations. Caroline Rowe is, as you can see there, Cytomics Data Science Research Manager at the University of Colorado Anschutz Medical Campus. She has a decade of experience in mass and flow cytometry, previously managing the Mass Cytometry Center of Excellence at Vanderbilt University. And as an ISAC Shared Resource Laboratory emerging leader, she has been instrumental in advancing best practices for high-parameter cytometry and data analysis. Dr. Celine Lages is an assistant professor at Cincinnati Children's, where she manages the research flow cytometry core facility. She supports hundreds of investigators each year in that position, with experience spanning immunology, hepatology, and multi-species cytometry applications. She specializes in panel design, cell sorting, and data analysis, and she, too, is an ISAC SRL emerging leader for her contributions to cytometry and core management. Let's get started. So, I wanted to start this with a summary of what we'll be going over today, which is to showcase the tenets of good cytometry data analysis. The structure today will be, each of us will be going over a portion of good cytometry data analysis. And in fact, a lot of us think about data analysis at this stage where we're trying to optimize our approach, but really there's importance in even before running your experiment and during your experiment, because if something goes wrong at those stages, it can show up in that data analysis. And today we're going to go over what that looks like. For our before section, we have Caroline Rowe.
Hello. So I'm getting a little reverb. Do we think that's just the echoing of this large room? Okay, thanks. Oh yeah, I should advance. So there's this concept I'm trying to get to catch on called pre-analysis, which is the thought that you need to think about your analysis strategies and goals well before you've even collected a single sample. And so I'll kind of go through components of this pre-analysis, this experimental planning, over the next couple slides. But ideally, all aspects of your experimental design and execution should serve the data science plan. And in turn, this data science plan needs to address the overall goal, the biological question, the objective of your study. So some key components, before you've even generated any data, you need to think about your sample type, collection, how you're preparing samples and storage, your panel design, how you might be modifying or updating your panel, and then how you're going to batch your samples, right, especially for larger studies, and collect your data. And so this is something I dealt with a lot in my previous role running the mass cytometry shared resource at Vanderbilt University, where I like to say we did everything from the tissue chunk to the final figure for publication, right? We were really involved in every step of what I like to depict as the wheel of pain, this kind of giant cycle. This example is very much tailored for the type of translational clinical adjacent work that my group does. But you could substitute in mice for people at any point in this, and any technology for your mass cytometry depicted here. So we were really intimately involved in every step of this process, from meeting with clinicians to talk about the goals of their clinical trial and whatnot, to collecting the samples, staining them, acquiring the data, analyzing the data, making figures for the publication, publishing the study, and then moving on to the next study. And please interrupt me at any time, or if I'm going too fast or you can't hear me, please raise your hand. So I've tailored this wheel of pain, again, a little bit for today's discussion. And in the upper left-hand corner, I've really highlighted the key questions of data analysis that you need to keep in mind at every point throughout this cycle. So we have this kind of ongoing joke between us that there's only really like 3 or 4 data analysis questions. Everyone thinks that their study is unique and special, and they're asking something in a new way. And really, you can kind of bin most studies, even the most exciting, into one of 3-ish categories, at least in the case of flow cytometry. So you're interested in something, right? The most classic type of question would be, I'm interested in a population of interests, like Tregs. How many are there? What do they look like in these samples? Maybe what are they doing in this new disease context? Or you might not care so much about specific cells of interest, but rather global differences, either over time, say one patient as they're receiving therapy, or between groups. Is there a way we can look at, say, the blood of patients who respond and don't respond to immunotherapy, and predict who will respond or not? And then kind of 3, the most squishy, exploratory, kind of the unsupervised clustering or trajectory analysis. More of a holistic characterization of the system that you're looking at. I don't personally love trajectory analysis. I think it assumes a lot, but can we say something about this new biological context based on prior knowledge and then examining new cells? And so it's really important to keep which of these 3 you're interested in as you go through the whole process of collecting data. Because even in the beginning, the number of samples that you're going to collect, the type of sample really matters. The quality of the sample really matters for the downstream assay. The panel you design has to address your biological question, right? Or at least approximately. There's some risks to over-tailoring your panel that I'll talk about later. And then finally, once you have data, the quality and quantity of that data will determine what kind of downstream analyses you can do. I've just shown a couple examples of stuff we usually do, finding changes over time. But whatever your question is, the tools need to address that, right? Like if you're interested in how many Tregs there are, then running a flow sum on your data won't be super helpful. And then finally, I like to think of statistics and the statistical strength of your finding as a separate analysis, right? We have our high-dimensional tools that we use to find novel cell types or characterize these high-dimensional data sets, but then the statistics is a separate after analysis process that will then inform your further studies. Cool. Okay. This is, oh, the timer's not going. That's brutal. Okay. So I could talk for days. We could have like a whole ABRF just about high-dimensional panel design. As a mass cytometrist by training, it's something I've been thinking about a lot longer than most people. And it's something that I'm pretty accustomed to explaining to data scientists and thinking about not only in the front-end way of like the biology of every marker I'm choosing and how they interact, but also how do I analyze them. And I think it's really helpful. Again, I love binning things. It just helps to discretize things. Oh, the timer's going now. Okay. Sorry. So generally, when we think of high-dimensional cytometry, at least I think a lot about immunophenotyping. I think about how do we tell what kind of cell the cells I'm looking at are. But then we can also, if you're measuring 40 or more things in one tube, we can also look at cell-cell interactions. How these cells that you're phenotyping interact with each other can be informative. A favorite of my group, and something I love because it's often cell type agnostic, would be signaling and function. And as we build bigger and bigger panels, I would encourage people to think about including cell type agnostic markers, functional markers, we sometimes call them, markers of proliferation, life, death, happiness of your cells, can be really informative across the broad context. And then the fourth panel here is microenvironment. This would be all markers on cells that are not necessarily the cell you care about, right? If immunophenotyping is one end of the phenotyping spectrum, then what's going on around those cells. And running a core for many years, I think a lot about what makes a good panel. And just briefly, it should phenotype all the known cells of interest in your sample, and also provide information on unknown cells, even cells that you're completely not expecting. And this can come in the form of non-phenotypic readouts, like I mentioned. And then finally, as we spend more and more time doing high-dimensional study design and collecting high-dimensional data, you need to have some kind of flexibility in your panel, right? The Cytof people were used to it, but dropping $10,000 on a full panel of antibodies every time for every different project gets to be rather expensive. So if you can have one mainly bulletproof panel that works across a broad range of biological contexts that you can maybe tailor a little bit for specific questions, that's a lot more cost effective and also makes your data analysis a lot easier. If you're used to seeing specific markers, you're used to how those antibodies perform, and kind of the information you can glean from those. And then, so we'll look for multiple sample types. And then along this, I also really like when you do have multiple panels to be able to align those panels can really help you increase your breadth of knowledge, right? And I'll show an example on the next slide. And then for those of you who are just getting into this, I would urge you to not just build your own panel from scratch. It's okay to copy. The OMIPs are out there, they're great. My favorite panel is the most boring panel that I've seen 1,000 times that I know works well, so that when I apply it to some exciting new tissue, I can be confident that the cell types I'm seeing are real because I have much, much data on this. And also in a core setting, I know because this came up more than once, keeping your panel very consistent over time can be super helpful when you say, hey, you have an unhappy customer who's very sure that their cells were alive and high quality when they gave them to you, and then you killed them, and they look terrible. You have longitudinal data to indicate that, no, this is how the panel works. It works well. And we've collected data with it before. It's clearly your sample. Not that you should have to do that. So this is I'm gonna grab some water. I'll let you stare at this for your favorite CD marker. Oh, it's the desert, it's so dry. So now I'll be kind of interweaving examples in here throughout. I'm talking a lot about immunophenotyping, immunotherapy, clinical trials. That's because that's what all my examples come from. But of course, they're applicable across a broad range of biological studies. So this is just an example 45-dimensional panel that I built for an immunophenotyping study. It's mass cytometry, which makes it easy, but I've heard 45 dimensions is well within the range of most spectral instruments now. And so I'd like to highlight two key things about this panel. On the top, in that gray box, we have our lineage markers. And this is your CD45s, your 4, your 8, your 14, 16, 3. So this panel was designed for immunophenotyping across blood, tissue, anything human. And it aligned with 3 or 4 other panels that my core offered that did deep phenotyping of other different cell types, but kept the same lineage. So we were able to align data across the panels more easily. And then at the bottom, I've got my cell type agnostic markers. We're measuring DNA, which I think everyone should do, Ki67, membrane permeability, cleave caspase 3, FAST, some things that tell you how happy the cells are, what are they doing. And these readouts can be really valuable, again, across a broad range of contexts. Even if I'm running this T-cell specific deep phenotyping panel on tissue, I can still learn a lot about, say, if it's a tumor, the tumor cells in that tissue based on these cell type agnostic markers. And then I have a whole host of other things I'm measuring all the way across. One thing I'd like to point out with these and with high dimensional panels in general is even though I've bin things here because I love binning things, don't think that because I've included CD45 RA, that I'm only doing that to measure naive T cells. It's also super useful for helping pull out maybe your NK cells. I think it's NK cells, RA, RO, NK cells, monocytes, nailed it, yes. You can kind of infer other cell types based on this larger number of markers without building a panel that's, say, specifically dedicated to detecting NK cells. And I'm happy to talk offline about non-traditional immunology, as I call it. Cool. So once you've designed your perfect panel, or that was a test, you didn't design the perfect panel, you're using one that you've already used 100 times before, you really need to think about your study design. As we move into high dimensional cytometry data, I'm sure some of you are starting to notice that it's really easy to pick up batch effect when you're measuring 45 different things. It was like one thing when we were just trying to figure out how many CD4 T cells were in a sample, and they're very robust and they survive things pretty well. But you can see some pretty striking changes based on sample handling, sample collection, all of that. So when thinking about data analysis, it's really important to plan the batching of your samples if you have a large enough study where you cannot collect all the data in one day, to kind of minimize as much as possible that sample to sample and batch to batch variation in a way that works with your data analysis strategy. And that's really kind of nebulous, but I'll walk you through this example. So for that immunotherapy study with my 45-dimensional panel before, we had 32 patients with 2 time points each. We had a pre and then an on therapy. So we had 64 experimental samples. That's a lot for us. For mass cytometry, it's a lot of time at the machine. And so the first thing you need to remember is that when you're doing this change over time or anything where you have multiple samples from the same patient, each patient is their own control. So they have to be stained and run together so that there's absolutely no technical variation between the 2 samples. Because I'm sure you appreciate between intraperson heterogeneity is huge in the blood and every other tissue, but we look at blood a lot. So first, we designed our batches so that every patient was run on the same day as themselves. And we accomplished this by doing viably cryopreserved samples. I know the mouse people like to do really fresh tissue a lot, and it may not be possible to do viably cryopreserved with every study, but it is helpful for planning. And we chose not to barcode because this was a clinical study and we were getting blood from patients in the clinic. We couldn't be certain of the sample viability, and especially with mass cytometry, but also with fluorescence barcoding schemes. If you have too much cell death in one sample, that can really mess with your barcoding across the whole cohort. So we did the high-tech tube coding, as we call it, where every sample is in its own tube, which takes longer, but for precious samples where you can't get another tube might be the best choice. And it also is because this whole thing took like 3 years. So it wasn't really viable to, we had to wait to get enough samples to run. So we settled on doing 5 batches of 6 or 7 patient pairs. And then, so those are all in orange and every patient is paired with themselves. And then the green tube is a customer-provided apheresis control, so a healthy-ish, PBMC-ish control from the customer that they froze and they split into 5 tubes and froze, so that we'd have 1 tube of exactly the same cells for every batch to kind of account for any staining variability or that sort of stuff. And we also asked, we were blinded to outcome, but we asked the customer to make sure that every batch contained a variety of outcomes, right? They had responders, non-responders, and then some like intermediate category in their study. So we were like, don't put all the responders in the first batch, because if there's some technical issue with that batch, that would be really sad for us. And then in addition to the green apheresis control, our core liked to do something we called the cover your butt control, which is the blue tube, which is a healthy blood from a lab member that was prepped and cryopreserved and then thawed and stained with the first batch, just so that if all the tubes that the customer gave us were entirely dead, we'd have 1 sample. We were like, well, we got cells there because we know that sample was handled appropriately at all points, because that has come up more than you would think. So it was worth it to us to like invest in having healthy controls around. And then it's cool if you do that with every study, then you have like longitudinal data on healthy controls on your instruments with your panels, which can be really valuable. And then, so yep, each batch was thawed, stained, and acquired together. The one thing I messed up was I did the antibody cocktail fresh with each batch, and this was mass cytometry. I could have prepared it all ahead of time and pipetted it as one thing and minimized some of the technical variation, but you learn. And then, so of the 70 tubes run and 32 patients, only 25 of them had enough data for first pass analysis, which I considered 5,000 cells per cell, 5,000 live intact cells at each time point. And this is a pretty standard outcome. People are like, oh, that went terribly. You lost like a sixth of your patients. But I was actually pretty happy with this outcome. So it was pretty on par with other clinical studies I'd run. And then, am I good on time? So this is what the data ended up looking like from my very carefully thought out batching scheme for this large study. So these are those apheresis controls I mentioned, the kind of PBMC-ish cells that the customer gave us. And so I have some biaxial plots in the top row, where I've got CD45 on the X and CD3 on the Y. So we should be seeing some CD4 positive, CD3 positive T cells, and then a bunch of CD45 positive cells that are CD3 negative, but not T cells, because it's PBMC. And what I think you can appreciate pretty well is there's some variation, right, in the level of CD3 staining, even though it should be identical, right? These are the exact same sample, just aliquoted 5 times and then thawed separately. But batch 2 looks a little bit different because I forgot to pipette in CD45, which is actually less of an issue with a 45-dimensional panel than you would think. It was more I just like chuckled. Actually, the most embarrassing part was having to tell the customer, but then being like, no, but it's fine, like the data analysis will be okay. So I had to drop CD45 from downstream analysis, which is fine. Cytoph has pretty robust cell detection. We measure DNA, we measure a whole host of surface markers, it was fine. But what is not okay is for that same data on the bottom, I have some OPSNI, technically, t-SNE plots. So and this is who's familiar with t-SNE? Some people aren't. Awesome. Okay. So t-SNE is just a dimensionality reduction tool we use regularly. And in these plots, like in the biaxial plots above, every dot is a cell, and I have contours to indicate areas of many, many cells. But the t-SNE plots are 2-dimensional representations of the similarity of those dots in the 45-dimensional space. And I like to call these fingerprints for people, because every individual kind of has their own high-dimensional structure of their blood data. That was not elegant. But people look like themselves with blood, and so any difference in the t-SNE for these 5 samples, which are all the apheresis control and should look all identical, is very, very bad. And so this is just a 2-dimensional representation of many, many, many technical problems. You know, some things are staining high. Yeah, hi. How
many cells did you stain for yourself?
As many as they gave me, because it's Cytof and you lose half at the machine. And we would tell customers 5 million cells in a cryovial will make me happy. 1 million cells I can work with. Beneath that, you are probably flushing your money down the toilet. Yeah, for Cytof. Yeah. But these t-SNEs, I think, only have, it looks like more than 5,000 events, but it might only be that, because that was my cutoff for including cells in the downstream analysis. So yeah, so a much bigger problem with this study ended up being that every batch had its own staining pattern in like a high-dimensional, hard to track down way. And this is where having every patient being their own control became a really big asset for us. We actually developed a whole tool where you can compare a patient to themselves and detect the differences. And since the patients were stained and run together, they didn't have any batch effect between the paired time points. So yeah, it can be, and that tool actually ended up being super useful for everything we do, and it's a lot easier than trying to batch correct your whole batches and then compare patients to each other. But let's see. Oh, and before we, hi, yes. So this data, she asked, the question was, did the clustering analysis give you similar clusters? I like to say I'm post-clustering. I don't whole hog cluster my data. So we run an algorithm called T-Rex usually. Because you could do it. You could put all the data together and run clustering, and there's enough, not phenotypic, there's enough technical difference, say, between CD4 T cells across batches that you're going to get different clusters for them on this. It's not accurately capturing the biology, right? You would want one cluster that's all your CD4 T cells, and then you could tell me how they're different across patients or time points or what have you. So we actually do a lot with KNN, k-nearest neighbors. So every cell becomes its own little cluster, and then we can determine if they're enriched in one time point or the other. And then once you've thrown away all the stuff that's not different, you can cluster on only the things that are different, and that's very easy to cluster. So I'm sure it'll come up later. Ask more questions.
So now that you have a good experimental design and a good panel, what can you do during your acquisition or around the time of acquisition, and what you should really think about when you start it? A big part is about the consistency of your data structure. It will help you at the end when you are really analyzing your data. We will also discuss a little bit about compensation or unmixing. I'm more a spectral person. I don't do Cytof. I learn about Cytof and what you should consider for this kind of data. And also the basic robust cleanup that you should apply before you even do high parameter analysis. So by my experience, when I get data, one thing was really worthy for me is to have a file name that I can come back to easily. So putting maybe the date of acquisition or your user or something like that is really helpful. The sample annotation and keywords are crucial because if you don't put them at that stage, you will have to go back and redo all your analysis because all this data cannot be used to tease which sample you are looking at. So always think about your subject ID, maybe the health condition, the treatment, the time point, the cohort if you have a cohort, the batch or things like that. Any information that at the end you could use to tease the information. And also the marker annotation, it really highly recommend your user to put the name of the marker and the fluorochrome. Because when you are going to analyze, going back and forth to their notes or contacting them can be tricky, and you don't want to lose time just to get this simple information who could have been incorporated when you acquire your sample. So really insist on that with them. It will help them also in the future. And also, you need to share your data, and it's nice for the people who are going to use your data in the future to find all this information. So, about unmixing and compensation, always think about, of course, I mean, we are I'm in an SRL, so I always think about make sure you have single color control for each of your batch. However, sometimes it doesn't happen. So make sure that when you are using your single color control for spectral, you really look at the signature, and that the user, when they are acquiring, make sure that what they think is in their tube is really what they can see on the spectral instrument. And make sure that the signal is always as bright or even brighter than your sample. And sometimes that is underestimated. If your single color control is less bright than your sample, your unmixing will fail. So there is nothing, I mean, you can do after maybe manual compensation, but not recommended when you have a 45 color panel. So really think that it needs to be as bright or brighter. And really look at this single color control and take the time to go through each one of them. Some maybe unmixing can be corrected if you really define properly what's positive and negative. So in general, on a regular flow cytometer, we were like taking everything who is positive, everything who is negative, and calculating the compensation. With unmixing, sometimes just taking the half of the brightest and half of the really, really negative will improve your unmixing. So that's something you can test. Maybe not during the acquisition, but later on, but really around your acquisition. I highly also, like I told you before, you can tell it's really important, make sure that the signature is really what you are expecting. And also, to check your unmixing, really take the time to go through your N by N plot. Everybody know what's an N by N plot? Okay. Some people like also N by 1, but you have more plots, so it depends on you. But really go through all of them and really look at the autofluorescence of your unstained sample when you are doing that. It will help you, and here there is an example of an unstained sample, and you can see, I cannot, which color is it? I think it's Alexa Fluor 488, and these cells were specifically positive, even if it was your unstained control. And also look at anything that is not really relevant for the biology, and one typical example that I can see, and here it's a paper who is published, so you can have access to it. It's this like little island who doesn't make sense, and in general it's aggregates that can be easily fixed before acquisition if you centrifuge your antibody before you pipette, but people don't think about it, and they can easily be removed. So that will help also to have a better unmixing. So after I've worked on your unmixing, oh, yeah, an example. So maybe, I don't know if we do. So here, for example, that was a single color control. On top, you can see what was expected, the signature expected for this fluorochrome. Is there people not familiar with signature in spectral? Okay. So in general, for each fluorochrome, instead of looking at only one channel, you are going to look at all the channels. And a fluorochrome is not only excited, I mean, it can be excited by several lasers, and you will have re-emission on, so you have a peak emission in one, but that's why you do compensation. It's also spreading around. And so for that fluorochrome, you will have all the laser and all the channel, and it's specifically for that fluorochrome. And on the bottom is an example of a single color control from a user who was using BV711, but have, I think it's obvious, a really different signature. We could discuss what happened. I don't know if somebody have like some suggestion. Maybe not. So in general, what happened, the most common things is you may have contaminated your single color control. And I think here it's maybe the case because BB711, you can tell that the observed signature is really brighter than what was expected in some channel. And it's BB711 being a tandem, I will have expected to lose the second part of the fluorochrome, but it's not what's really seen. So it could be that. There is maybe other explanation, but that's the one who comes the first in my mind. So in that case, if it's really just a contamination of the single color control, you can just maybe rerun a single color control, and that will help you to do the unmixing properly, because you cannot use that single color control to unmix. The manufacturer, so here it's a Cytec Aurora, so the manufacturer will provide you every signature for all the fluorochrome available. And there is a lot of new fluorochrome coming on the market, so they are not always updating, but you can make your own signature, and you know how your fluorochrome behave, and you can always compare. But you can tell them, and they will do it too. And they are good at updating also. How I introduce that. So so you are really happy with your unmixing. There is a step that you can use to really clean up your data. And here, on the right side, it's current protocol. It's a really good paper because it really gives you step by step what you should do. And it covers a big part of my part of the talk. And they are showing you, so you have a t-SNE, and they go through different gating and look in the t-SNE when you are gating on a specific population where it's located on the t-SNE. And you can see the first cleanup they did was a time parameter, just to look at how the acquisition was performed by the instrument. And you can see they removed the last events during their acquisition, and that gives you some islands. Let's say, call it island. I think it's a UMAP. I cannot read, so I think it's a UMAP. So it's a kind of an equivalent of t-SNE, just distributing and clustering the data a little different, but same idea. Everything who is in gray is the population they remove from their gate, and the red is the population inside their gate. And you can see some population, some island will not be selected if you remove this not smoothly acquired part of your sample. They did also a doublet discrimination, and you can see a lot more more island disappearing or not being used for your data analysis. They did a second doublet to make sure they really have only single cell when they are going to acquire. They did after, they removed their aggregates, so maybe not really visible, but the aggregates in that plot are in red, and it's a really small island. So you could use that also to remove them, but you can tell, like, if you didn't remove these aggregates, if you are comparing sample to sample, and some have aggregate and some have not, you will think that a population is specific for a sample. When it's not, it's just in that sample you have aggregate, and so you are overestimating, like, maybe the impact. And they continue their manual gating by looking at gating just FSC, SSC, so just the size and the granularity, and now it's really dropping. You can see there is really few islands who are really of interest for you, and gating really on the lymphocyte. So it's a PBMC, so you can distinguish lymphocyte and monocyte, and just by gating on the different population, you can see where are your lymphocyte or your monocyte. There is now a lot of automatic cleanup tool. I'm familiar with FlowJo, so for me, I have access to 4 different plugins. You can test them. They have different cleaning methods. Some are more restrictive than others. So in function of the number of cells maybe you have acquired, you will prefer one versus another. In general, it really depends on the number of cells I have at the end to analyze. So after cleaning up this data, there is a process that is often underestimated. It's the scaling of your data. So on the top, you have You have a sample where the cofactor applied for this scaling was 5, and it's maybe too far or not clear, but you can see that they make two different rectangles. They are based on the fact that, sorry, I should say it differently. What happens if you don't scale properly, the most often it's your negative population will start to split in two. And if you don't pay attention to this, you will think that you have two different populations. On the top here, they show in blue, I can really not read. So the blue population, it's the first rectangle, so the one who are really below the 0 of the negative population. And the orange rectangle is the one who is a little closer than to the positive population. It's not that you have really 3 populations, it's really just the split of the negative. And you can see on the UMAP, you have like completely your data clustered differently. So by correcting that and making sure your negative population is only one population, even if you split and try to see the really negative versus the a little bit positive in your negative population, you have exactly the same location on your cluster and not overestimating or thinking that you have a new population created. I'm not sure I'm clear. So really look at your axes. If your negative population, if you have really extra negative population, you should start to think about the fact that you are splitting your negative population. And that will impact your data. So for the scaling, yes, it's in general. I mean, it's not even a plugin. It's already inside the software, and you can adjust if you want. Yeah. So I don't know if we have time because Alice have a lot of nice trick. Okay. So here is an example. You have somebody who come to you and tell you, I am looking at T-Regs. We really like T-Regs in this group. But my T-Reg are only in my CD8. So what happened? Is it like normally T-Regs are only, I mean, mainly in CD4 T cell, but they could only detect them in the CD8 population. And that, I wish we have a laser. Oh, we do. So here it's CD8, and so that's the island where the CD8 should be. Here it's their FOXP3, and you can tell it's overlapping exactly to the CD8 population. So the user is asking, so what happened? Why I'm not seeing it? So do you think it could be something associated with the data structure or the compensation and mixing and scaling, or because the user didn't clean enough their data? What do you think? Who think it's 1? Who think it's 2? A little bit. Who think it's 3? Okay. So in fact, if you really have all the plots, you have easily your answer, because in fact you can see here, when you look at FOXP3 versus CD4, the unmixing was not appropriate. But when you correct that unmixing, now you can see your FOXP3 positive CD4, it disappears. And your FOXP3 behave exactly as your, I mean, at least in the CD4 population, and they are also expressing CD25, which is also a criteria for characterization of FOXP3, of Tregs. So really take the time to look at their unmixing and really check. That's why I was insisting about the n-by-n plot, because that will tell you really quickly, you will see that and will like, yeah, there is something that is not right. So go back to your single color control, try to re-unmix, make sure you take the extreme positive, extreme negative that may correct. You may have a problem with your single color control fluorescence, the signature not being appropriate, so you could reacquire a single color control, for example.
Great. So now we're to considerations for selecting a best analysis approach or tool. And this is normally where we start. I'm so glad we got the preface from Caroline and Celine here, because really a crucial part for selecting an appropriate tool is that, one, it addresses the question. And of course, you guys have considered this thoroughly, plotted it out, done excellent experimental design.
I definitely didn't just collect gigabytes of data with no plan.
And additionally, you acquired your data very well and have cleaned it up and processed it such that it's ready for analysis. So you don't have batch effects or unmixing issues or sample quality issues. What I'd additionally add for selecting a best tool is that when you're using it, you're aligning with biology, so you're doing your appropriate scaling. Celine showed how that can affect your results. You're using the relevant analytes for your question. You're not just throwing in every single parameter. And you're getting out the biology you'd expect based on your question, which you've already defined, in that you're getting your expected number of subsets, and they're expressing what you anticipate. The last consideration, which I'll mention here, but is very personal to you, is in selecting a best tool, it needs to consider your resources and your needs. So how competent you are with the platform that you want to use, your time, dollars, and support, your computational power, and your need for flexibility and control. And I'll go into a little bit here. Oh, sorry, I guess I'll do this first. I want to mention before I go on that a typical analysis pipeline has 4 stages. So I get confusion oftentimes when people say I'm doing flow cytometry analysis. There's really kind of 4 types of analysis. I don't know why this is animated. Okay, weird. All right. Acquisition, some people call analysis. I would just call that acquisition. Then there's pre-processing. This is where you do all your cleanup, compensation, debarcoding, batch correction. Then those cleaned up files then go into what I would consider flow cytometry data analysis. This you could apply dimensionality reduction techniques, population identification, either manually or by clustering. And then that creates values, which Caroline had already suggested, then can be transferred over for statistical analysis for data insights. And it's important to do those pre-processing stages and experimental design so that you have good quality data coming in. And then on the analysis side, to know your question and understand the biology going in. And I will mention that computational and algorithmic analyses can be applied at all of these stages, but they don't have to be. And in fact, there's no necessarily right approach when you're doing analysis. Find out what works for you, again, according to those guidelines that I set out. And some tools that are available may bundle some of these approaches, like when you're looking for differences in populations, data analysis and data insights. The statistical analysis may be combined. What I will say in terms of resources, that there is a lot of analytical tools and platforms out there, and they vary by ease of use. So I've kind of arranged these on the left-hand side. You have the most options, they're free, but they lack support. And on the right-hand side, there's the fewest options available, but they've been well developed and have full support, also more costly. So you can get programs if you go to the code. You can go to our code-based algorithms. I've put some links up there. I think I have somewhere on here a link to a PDF version of those where those links actually work. Some common ones, FlowSOM, Peacock, CytoExplorer. The level kind of where I'm at more is the software plugins. So FlowJo, for instance, has FlowJo Exchange or FCS Express, where you can pipelines where you can download these algorithms and use them in a comfortable setting for you. The next step is these cloud-based platforms that actually house these. Examples being Cytobank, Omic, and Tursin, where you can kind of generate your own pipelines using already well-vetted, basically R scripts. And then finally is the full analysis pipeline, where there's some examples down there, Astrolabe, Ozette, or Gemstone, which you can put your data in and get data analysis out. That's been curated for you, and you have your handheld but have a little less insight into what's going on. And you can switch between these platforms depending on your needs for any particular question and for different parts of the pipeline that I've just shown you. And there are no necessarily right tools. And for the sake of the time here, I'm not going to go through a bunch of tools and which would go well with your project. But you can sort that out. And I will say that, let's see what I have on the next thing. Okay. I will say that the main obstacle that we found in a survey to using analytical tools was their uncertainty as to whether the tool would answer their question. And in living that reality, I sort of feel it's less about understanding whether the tool will answer the question and more about whether they have defined their question. So I really want to say that if you go into data analysis with a common English question in mind, you're going to get a lot simpler results out. So here is where I really wanted some audience interaction. I've given 2 examples. Here's the first one, based on true stories, where you guys are going to help me diagnose possible issues in this data analysis. So this person comes to you after, collecting their data, clustering it, and they say, I clustered my sample, but I can't find the CD4 Tregs that I'm interested in. And so it happens to be I don't know if this one's a Cytometry panel, but it's a large panel, and you can see here, they've clustered on all the markers that were in the panel using human whole blood. And they say, hey, there's 2 clusters here, but I identified my CD4 T cells. They're expressing CD3 and CD4, but there's no CD25 high cluster within that. So let's try and diagnose this, keeping in mind the fact that a good analysis approach addresses the question, is appropriate for your data type or quality, and your approach aligns with biology. That's true. They were going to use CD25 and found that it didn't have a cluster. Exactly. I see you guys mentioning markers a lot here. It's 127 is in it. Sorry. Oh, boop. No, I'm. Oh. Great. So that's one option, is to go back to other Treg associated markers and make sure that those are all performing correctly. There's no right answer here. This is made up data based on true stories.
I'm sure they were excited to use this clustering algorithm, but if only there was some tool where you could look at one marker against the other marker and then say where your cells of interest were, that might be faster.
Are you saying their question of what are other Tregs is not aligned with their approach of just clustering? I think that's a great solution to a question where they used an approach basically that wasn't addressing their question. They had a specific subset in mind and then used an approach that wasn't particularly good for that purpose.
I know my experience with flow cell is the settings, you put the right settings to get the granularity right. And the clustering algorithm is too coarse, too fine, and you have to play with that setting of the clustering algorithm.
That's an excellent point too. This person is new to clustering and just went with the default settings. So you could imagine possibly getting more granular and getting to your Tregs.
I would also ask them if there's a reason they need to find these in a less supervised way, right? Yeah.
Great. Yes, exactly. One good way to get to better data analysis, especially when you have a target population in mind, is not to analyze all the other cell subsets. If you could clean that up, just do T cells or CD4 T cells, then you might get your answer. Great, so I think I heard everything that I had in mind, basically. We want to address a specific subset question. We used clustering when really maybe gating would be just fine, or some even automatic gating, where it's very targeted. For aligns with biology, again, too broad of a cell population was clustered. You might have much better luck if you narrowed it first to just CD4 T cells or T cells. And then we were talking about the markers that were used, or that could help reflect if we actually had clusters of Tregs. In this particular setting, all markers for a panel that was developed for function, you'll see lots of phospho markers and such, were used for the clustering. And that's really not appropriate if you're just trying to find a particular subset. And lastly, I mentioned batch effects might be limiting that distinction when you're clustering with all your samples together. In this case, I didn't say it was multiple human whole blood samples, but slight shifts in Treg defining markers would put them on different places in n-dimensional space that might just shift it so the clustering wasn't grabbing them. All right. Ready for the next one? This is another common theme I get. Help me. I'm going to publish this novel rare T cell population I found that's expanded in my diseased individuals. And you'll see on the left, a healthy control TISNE, and on the right, a diseased TISNE, and you can see there's this population that's only in disease, and when quantified, it's higher in the disease than the healthy control. And then also this population, when looking at the intensities of these different markers, you can see it's, CD3 TIGIT high, CD127 high. Very excited, going to publish this. Would you hold reservation for that publication? Or would you say, go ahead? Do you want to check something? Batch effects? Yeah. Presumably there's something expressed only in that. What if they came back and said, I put exactly the same amount of healthy donors and diseased individuals per batch? I'm just playing devil's advocate here. That's a great suggestion. Right, so it's not cleaned up well, not honed in on the subset of interest. Yes, because what I didn't say here is that there was no cleanup done on this particular sample. Right, the comment was if there was no cleanup done, we don't know about viability. We don't know what that cell population is. And it's a little suspicious that they're so high for some markers that aren't so typically expressed. Oh, right. And I have seen this exact kind of case come out where bioinformatics, it hasn't been communicated to them what the interest is, other than I want to find a difference between healthy control and diseased patients. And they will find things different between healthy control and diseased patients because they assume the data that you're bringing to them has been collected well, batched well, and cleaned up well. And so I just want everybody to be aware that that is not always the case. Right, so addressing the question, make sure it's batched well, identify, oh yeah, if you identify through an algorithm, I think that somebody said this, like manually gate to verify, go actually identify the population, making sure it's appropriate for your data type, have the cleanup steps been performed, aggregates and dead cells and instrument anomalies can all result in these extra clusters. And then whether it aligns with biology. So ensure the population expresses physiological levels of the marker. So it's very rare to see like subsets expressing that high of TIGIT or CD127. So this is where communication with the biologist, immunologist is really important. And is there a precedent for that population to exist? Do we have CD3 or do we have CD14 expressing T cells? Thanks for going through that exercise. So we've put together for finding a best analytical approach this diagnostic checklist. So if you do all these things, you are more likely to get better results in the end. And if you see data that you're not sure about, these are the points which we suggest, and please suggest if you have any more, that you can go back to understand whether that approach, that analysis is believable, is accurate. So as Caroline had demonstrated, before you even acquire any data, make sure you define your analysis goals. And I really love that you could really limit this basically down to 3 different questions. You either know a population that you're trying to find, you want to assess differences either over time or between groups, or you're doing an exploratory kind of clustering trajectory analysis. And it's okay to do more than one on a data set, but just know that that might require separate tools. A robust and flexible panel with optimized titers going in, and then appropriate experimental design, such as batching, and then including controls, so that if you do find something anomalous between healthy control and disease, that you can always go back and make sure that it wasn't just something in your panel or technical that day. And then as Celine went through, during creating quality data, having consistent annotations, accurately uncompensating and unmixing, and then the pre-processing. So this is all the cleanup, removing of aggregates and dead cells, making sure you have the right scaling, and then you may need to do a batch normalization, depending on what you're going in for the analysis. And then lastly, which is where most people start, unfortunately, is finding the optimal approach or tool for analyzing the data once it's been generated. You have to make sure it matches your analysis goals. You want a tool that's compatible with data quality, because even if you have batch effects, there's some tools that are more robust to that or not. There's combo tools that might clean up your data for you. And you want to make sure your analysis is aligned with biology. And I think this is something that uniquely should fall into the hands of the person doing the experiment when they're collaborating with the data scientists. You have to make sure you define which markers you want to have analyzed, as well as your expectations around number of subsets or expression of markers. And then, of course, make sure you have sufficient resources. I think we all struggle with that. And remember, as you're going into all this, you are never alone. There's so many great resources for data cytometry analysis, notwithstanding the group up here, which we've put our emails, so if you want to contact us, you can. Certain vendors, SciTech is listed here, have great resources, as does Cyto. Cyto University, which has, a wealth of presentations on the subject. Caroline, do you want to promote your?
Oh, yeah, and Celine showed a great example of spectral data cleanup, but my high-dimensional phosphoflow protocol is kind of a nice starter mass cytometry phosphoflow protocol that has a mass cytometry data cleanup, as well as like intro R scripts that you can download, and it has demo data that you can like run through the pipeline. It's pretty old now, so it's pretty rudimentary, tsne, flowsum, but it's an easy place to start.
sorry. I just wanted to mention ISAC is also really developing some videos to explain also the different algorithms that are available, like what's the difference, how it works, what they were considering. And it's really important that, I mean, for me, to understand what the tool is made for, because they are created for a specific question, and everybody else is using it, forgetting why the tool was created. So they are developing that.
Great. We can open it up to you guys if you have any more suggestions, want to discuss data analysis.
We can go back to specific examples if there's deep questions, but. So the question was, you're doing flow on meninges? Is that what? Okay, sweet. I do a lot of brain work too. It's super fun in a well-defined biological space. So something I see a lot, like, so we're showing really beautiful UMAPs and T-SNEs, right? Because we're doing immunophenotyping on blood, where like it's a single cell suspension already, and the cells are pretty happy, and we know what markers characterize them really well. So you're getting really beautiful separation. But what we don't show is all the T-SNEs I've run on glioblastomas, or on poorly defined cancer-y neural things where maybe the antibodies aren't as robust, the biology is certainly not as well known, and you're going to get way less separation in that context. You're going to get more balls and blobs with edges, and you're going to have to. So for that, I mean, if you have a specific population of interest, I would definitely go with biaxial gating to find it if you're just trying to quantify your thing. Beyond that, there's panel tailoring, which takes time and money, and but a lot of what and there are parameters you can tweak on TC and OPSNI and all that, but if you're running it through like FlowJo or some pretty standard analysis platform, it's likely not biology, right, but like limitations of the data set that you're working with, right? We do unsupervised analysis, but it is supervised because we picked markers, and if you are kind of just picking the best markers and they're not that good, yeah, it's just part of the game.
So, Caroline, you mentioned DNA staining.
No, go for it. I did mention DNA staining.
Yeah, so does that come from thinking about just from a liability perspective, or is there something else behind that, like looking at senescence?
Yeah.
Cycle in everything, right? Because mass cytometry versus spectral, right? Like thinking about, coming from the spectral side of things, if you could throw hopes in there or something, potentially if it works in your panels, maybe get that additional amount of information, it could be really quick. But especially in like a complicated sample, right, where you have a lot of junk and you just want to clean that up. But maybe you mentioned that for another reason, and I'm just curious.
Oh yeah, so we do DNA and cytoph because there's no scatter, so you have to find cells. But as I've been like getting more into spectral data sets, I do really like it as like just to really know that it was a nucleated cell and not debris, because I am doing a lot with brain, and we do kind of like we don't like clean up our samples much on the front end, so I really have to be able to identify the cells in the data versus the chunks of myelin or what have you. And I think it's something that maybe has been underutilized in the fluorescence field. These pan cell markers that would be helpful and increase your data quality. So it's my little soapbox for everyone. Yeah, that's really cool. So just to repeat for everyone, because it's awesome. So they're running a sample that's stained for the nuclei, and then using that to kind of identify, like, this is where our single cell area is, and then run your stain sample, and then you've kind of, that's really, yeah, that's neat. And then you don't deal with the spectral overlap into the, yeah. Real quick, I want to talk about the brain with you. But in the last 45 seconds, any overarching broad questions? Otherwise, I might break, and then we can just chit-chat. Good, cool. Thank you for your attendance. Thank you, Alice, for organizing this. Thank you, Celine, for being here.